Hello class, this is Mr. Mormon. Welcome to our 6th video in this series on genetics. Today we'll be talking about protein synthesis. Synthesis means to make or to build, to take small molecules, put them together to make a larger molecule. It's the opposite of digestion. So what we're going to be learning about today is how proteins are made. So why are proteins so important? Proteins are important to all of biology because it's basically what living things are made out of. There's structural proteins like muscle, bone, and hair. And then there are important proteins that are controlling all the chemical reactions that are keeping us alive. Those proteins are called enzymes. So it's clear that cells have to continuously make lots and lots of different proteins. Now, when we think of proteins, an image like this might come to mind. These are all protein-rich foods, and if you think about it, it makes sense that proteins would be in these foods because they are, biological living things made of muscle and other tissues. That's all protein. So lots of the parts of animals and plants are going to contain a lot of protein. Now, we need to eat a lot of protein and have it as part of our diet because we digest it and break it down into its little bits, the amino acids, so that we can build new proteins in our bodies. Now, recall that the building blocks of proteins are amino acids. Also recall that proteins are assembled by the ribosomes inside the cell, and the instructions are coming from the DNA. That's the genetic code that is used to build all proteins. It's also important to know that all proteins have a special shape. When they're put together properly with the amino acids in the proper order, they bend and fold into a special shape, which allows them to do their job. Here are some examples of different biological proteins. You can see that they all have very unique and interesting shapes depending on how those proteins, how the amino acids make the protein fold and bend into all these different intricate shapes. And that's how they do their job. Now, we've already talked a lot about DNA, and we know that that's where the information is stored. But now we need to know about RNA as well, because RNA plays an important role in protein synthesis. And there's two main types that we're going to talk about today. Messenger RNA copies the information from the DNA and delivers it to the ribosomes. Transfer RNA carries specific amino acids to the ribosome so it can be added to the growing protein chain. Okay, let's compare and contrast DNA and RNA. First, we can say that they both contain genetic information. Now, one of the main differences is that DNA is double-stranded. Remember, it's that double helix. It looks like a twisted ladder. RNA is only a single strand. Another difference is the bases. In DNA, the nitrogenous bases are A, T, G, and C. In RNA, there are no T's. The T's are replaced with U's. Finally, because DNA is double-stranded, it cannot leave the nucleus. The pores, the openings in the nuclear membrane, are too small for the DNA to fit through. RNA can squeeze through, therefore it can leave the nucleus and go out to where it's needed, out in the cytoplasm where the ribosomes are. The process of copying the DNA message into an RNA message happens in the nucleus, and that's called transcription. Ribosomes then take that RNA message and turn it into a protein, putting the amino acids together in the right order. This is done in the cytoplasm. Now, the genetic message is broken down into groups of 3. That's called a codon. It takes 3 nitrogen bases to spell out the information for 1 amino acid. And finally, there's the anticodon, which is the 3-base complement to the codon, which is found on a transfer RNA. The codon and anticodon will fit together like 2 puzzle pieces. And the ribosome's job is to find the right anticodon on a transfer RNA and plug it into place. Now, this is the very complicated diagram that we did in class of protein synthesis. Everything that I was just talking about is happening here. Transcription is happening here in the nucleus. The messenger RNA is making a copy of the code that's on the RNA. This single-stranded messenger RNA will then come out of the nucleus. It fits through that little opening in the nuclear membrane. It comes out of the nucleus where a ribosome attaches to it. The ribosome attaches it and starts reading the code 3 bases at a time. Now, the code that was transcribed, the codons are GAU, CGU, AUU in this example. So I wrote them down here at the bottom. We can now do the translation part where we take that codon and turn it into the right amino acid that gets put on. And what the ribosome does is this whole area around the ribosome is surrounded with these transfer RNAs. Here's one here, here's one here, here's one up here. Now, what a transfer RNA has is an anticodon at one end, which is a complementary 3 bases, which will match up with the codon when the right one is put in place. And at the other end is the next amino acid, which will be added to this growing chain. So here we see a transfer RNA that's been plugged in. This is the next one that's been plugged in, and the ribosome will then move to the right and look for the next one to plug into place. So I've already filled in here that GAU was the codon for this amino acid, and that's aspartic acid. The red one is arginine, and that was CGU was the codon, and AUU is the codon for isoleucine. So I've labeled those amino acids. But how did I know that these were the amino acids? Well, there's a chart for that. Okay, so this is the chart that you could use. The way this works is you start in the middle and you work your way to the outside. Now remember that first the first codon was GAU. Starting in the middle, here's G, A, U, aspartic acid would be the translation. That would be the amino acid that would be added. Here's a different chart than that bullseye-shaped one, but it works the same way. So again, if our codon is GAU, to use this one, you go kind of around the edges. So G, the first base is G, so it's going to be in this row. The second base is A, so it's in this row. This column, GAU, is right here. ASP is aspartic acid. This is the kind of chart they usually have on the Regents exam. Okay, the last thing I'm going to show is an animation from YouTube that I'm going to embed in this video, which hopefully will make everything that I just went over a little more clear.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis. Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the three-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.
Okay, well, that's how proteins are made. As always, if you have any questions, please ask them tomorrow in class.